Sri Gauri says she has often heard about a third approach called postbiotics
Mhm.
that skincare market targets. How is this approach different from the other two?
Okay, so what is prebiotics? Prebiotics is when basically bacteria are given something, let's say food or some conditions to produce desired molecules, desired metabolites. Probiotics, we understand that's when a live bacteria, or usually actually not live bacteria, but some bits and pieces of, good bacteria are used. And postbiotics is when it's actually the easiest approach, and easiest from the point of view of applications, because the good bacteria is grown in a lab, and then the product of this bacteria, the postbiotic, the result of their activity, a metabolite, anti-inflammatory molecule, whatever it is, it's this product that is used and applied, let's say, in a skincare product.
Oh, all right. That was quite helpful. Do we have any other questions? If yes, please put in the chat. And I have a general question that I would like to ask. So, when we look at different data sets, we find some data very specific to a specific condition where we don't have the control samples in the data set, right? But can this thing be done that if we have a data set, let's say for a condition, and we have a control sample set from a different data set, and then we would like to compare it? Is that possible? Because we were discussing this, that it is not actually that the microbiome stays constant. Like, is there a normal microbiome to which we can compare any data set?
Great questions. Yes. So I don't think that, we have this kind of standard healthy microbiome reference data set like we have in, let's say, human genetics, where there are some reference human genomes. And yes, there are certain problems with them and so on, but nevertheless, these are the reference data sets that everybody is sort of comparing their data with, right? We don't have that in microbiome. We don't have it yet, okay? I'm sure we will have it, soon. However, having said that, I am sure we can use the controls from other studies, because, we can use the reference data sets or controls from other studies, provided, they're the same with age and various other factors. So there are many factors that we need to take into consideration. Pollution, if these people live in big cities, what is their lifestyles and so on. But overall, I think age and gender obviously are the ethnicity as well, actually. But this is not really known whether the ethnicity is a factor, or whether, let's say Japanese microbiomes differ from, Western European samples because of lifestyle or because of ethnicity. We don't know that yet, but the general conclusions are the same. So yes, the short answer, we can use controls from other studies, provided we check for age, gender, and ethnicity. Does it answer your question, or did I miss something? Okay, so there was a question here, does skin microbiome change? It's a very deep question, actually, because yes, it does, but not, it seems not in the way we think. It's not sort of very fast changes. And the changes actually happen on the dermal level, on the more internal level, rather than on the external level. So there is lots of data on that. And again, I'm sure a lot more is coming, but it's not, there are certain transient changes, let's say you travel somewhere, you board the plane, and then after that you feel your skin is dry, and obviously you caught some bacteria. But if your skin is healthy, it will fight this kind of foreign bacteria and will recover to its sort of natural state.